didn't present myself. My name is Uzi Zohari. I'm a member of the LSEC, the neurobiology department here. I study vision as well, just the student of Shaul's, who you've heard a while ago. And what you're going to hear about from me is basically higher level vision, and specifically, I mean, what do you choose when you want to teach something like higher level vision? Of course, I mean, vision is a topic that you can give a whole course about. And here we are, I have only 2 hours after you've heard the basics from Shaul to cover one aspect of vision, and I decided to talk about object and face recognition. I'm sure there's much more here than we will be able to cover, but that's not really crucial. The one thing I want to make sure before we start is that we understand what the problem is. And I'm sure you went over it with Shaul, but just to probably repeat something that you know yourself, what is the problem with basically vision? It's the fact that originally there's a representation. The truth is, this is the first question I ask any student or any person I meet, when they ask me, okay, what do you do in your life? Okay, I study vision. Well, what's so interesting about studying vision? That's a good question. But the main thing is that the problem is not always clear to the layman. Of course, the problem is clear to you, I presume, and that is, okay, that vision, initially there's sort of a transition from the world out there to a representation that we have on the retina. But the representation on the retina, of course, is basically just light and dark patches, which are then translated into electrical activity. And of course, the brain's job is to make sense of these patches of light and dark, okay, in color as well, and to make sense and to figure out that there's a class next to me now that is waiting to hear what I have to say about object recognition. So, of course, parsing this visual scene into objects and figuring out the relationships between them, that's all about brain function in terms of vision. And as you may know, something like 1/3 of the brain of the primate and the human is devoted to visual analysis. So, one thing is, again, to make sense of it, but there's another thing which you will hear a little in one of the talks of the students in Wednesday, and that is, okay, it's not only just to make sense of it, it's to make sense of it when the illumination changes like this, okay? So now your faces are illuminated completely different, and the source of the illumination is from such a position that I'm sure, as you probably know, in computer vision, this is a very serious problem. I mean, just to be able to extract what the images, what are the objects there, when there's changes in various aspects of the visual image, such as illumination. What else? Let's hear from you. Okay, so distance. Of course, as I'm getting closer or further away from an object, the retinal image becomes bigger as a function of our distance. Right, so distance is another constant. Rotation. As I'm moving, like watching you from here or from there, of course, the image is rotated, or view are changing, okay? So rotation would be another. Okay. Translation and rotation. Okay, translation is an easier problem, but yes. What else?
Objects might be partially covered.
So, yes, occlusion issues, issues of occlusion. That's another. Anything else? Scale. Okay. So scale has to do something when we talked about distance. That's part of it. Yeah. So the conversion, the fact that the retinal image would change dramatically as I'm getting closer or further away from you, and so you have to learn to scale the retinal image and to extract from it its true size, its physical size, irrespective of the retinal image. You take into account the distance. Yes?
Angle, decoding different angles of the same object?
Yeah, so we mentioned that before, the fact that, you could translate or rotate it the way you're looking or the object itself may be moving. Okay, so we may be looking at it from different perspectives. And of course, something related to this, illumination is this issue of not just the image, the energy returning from the image, but more, which is a function, of course, of the illumination. Okay, so the illumination, first of all, in terms of the spectral contrast of this light is very different from the sun, but also the issue of shadings. Okay, so of course, if you are to measure with a photometer an image, and now you change the illumination of the direct illumination, of course, the shading would change accordingly, and therefore the physical output that you're going to get from this photometer is going to be completely different depending on all these parameters. And we are able to discard these, and we are tolerant to these changes. And of course, this is not a trivial problem, as people in computer vision have learned the hard way in the last 20 years. Now they're getting better at this, and I don't know if you've heard anything from people who are doing computer vision, but I suspect that most of it is just brute force because, you know, the computers have improved so much in terms of their computing power. So that's, I mean, algorithm is part of it, but clearly computing power has a lot to, has contributed a lot to the fact that they're much better nowadays than they were 20 years ago when I started my PhD. Anyway, so, but this is a physiology course, so I'm going to basically stick to physiology. Illumination, okay, there's some situation where illumination does become somewhat more difficult to compensate for. And the first thing I want to start with, let's see. So welcome. Okay, back to low-tech. That's okay. Here's the low-tech, my low-tech style. Okay, so have you seen this from Shaul?
No.
No. Do you recognize what this is?
Monkey brain.
Monkey brain. That's true. That's the monkey. By the way, this is the eye, and here's the monkey brain. It's a view. This is a view from the side. It's called the lateral view, and this is sort of when you take the two hemispheres and you look from a medial view. So just to give you an idea, look at how big the eye is compared to the brain. This is 1 centimeter, okay? And what you're seeing here are, in color, are the areas devoted to vision. You see that there's multiple areas, and this is what's called a sort of flattened brain, sort of an atlas representation. So you've covered with Shao the retina, I believe, and the LGA, and you've gotten all the way to V1, and now we're going to talk about all this. That doesn't work very well. Okay, sorry.
The pointer.
חשבתי שהפוינטר בזה. That's, okay. That's yours? That's your personal one? So that.
Turning on the side, on the left side. It's turning on, not on the side. Yeah.
Okay. Thank you, Ben. Okay, so what you see here is just sort of a picture which is not up to date. I think this is from something like 12 years ago from Vanessa, showing the multiple visual areas that have to do with basically visual analysis. And the first thing to notice is that there's over 30 of these areas. And there, as I said, about a third of the brain is of the monkey. This is the rhesus monkey brain is devoted to this. And we'll see in a minute. There's some division between these areas, but on the whole, how do you define an area? I mean, why did they say, okay, this is one area, this is another area? Any ideas? Yes.
Depends where you're from. If you're from psychology, then by your anatomy, but it looks like physiology here. If you're from molecular, then proteins express the neurons.
Okay, what about neurons? Okay, this is electrophysiology here, so it's not, okay. Initially, לא שומעים אותי מה?
That's for the recording later.
It's the recording later. Okay, sorry about that. run into recording myself. Sorry about that. Okay, not that we covered much, but here we are again. Okay, so how do you define an area from the physiological perspective? Okay, neurons fire in some way, and they should be visual. But how do you decide? Okay, so you find visual neurons here, visual neuron here, and visual neuron here. Morphology is the same. If you were to look at, from, cytoarchitectonics of the brain, you would not be able to tell one area from another in general. Although Brodmann, of course, there's the division of Brodmann's areas, but that's not Brodmann's areas here. Any other ideas? Functional. That makes sense. Okay, what about functionality? Not think out loud. I mean, what would be? Okay, so you've heard. I'm going back to what you've heard from Shaul. You've heard about maps from Shaul, a retinotopic representation of the image, meaning what? That if you are to record from individual neurons, and you're going from one neuron to the next to it, they're visual neurons, so they have a receptive field, right? You've heard that term, the receptive field. And what about the receptive field? They should be overlapping. If you record from one neuron, you go to the next neuron, the receptive field tend to be overlapping. So the important point is that there's a mapping, okay? It's not that one receptor, the first neuron has a receptive field here. Let me draw this, it'll be easier. So yeah, there's the fixation point. Here's the receptive field of neuron number one. Okay, here's your electrode. It moves in, records from here, number one, moves on, records from number two, number three, and number four. And generally, just to make the point, the receptive fields would be overlapping. It wouldn't be the case that number one would be here and the receptive field of number two would be here. No, they form a map, okay? And the idea is that as you move to the next area, there's another map. So there's a duplication of the world. And, okay, the duplication terms that each area has its own map of the world. But not only that, each area, we tend to think, does something a little different than the other. Now the question is, why would you need 30 areas or 30 or so areas in the visual system? So they do something a little different. And we can go on to the next slide to think about what do they do. Okay, so here's sort of a scheme showing the connectivity between the different areas. This will be in the test, okay? No, it will not be in the test. Okay, so it gives you an idea. Okay, 30 areas, look at the connectivity scheme. So it's not that everyone's connected to everyone. Everyone, first of all, the information does get from the retina, and to this is V1 here, okay? And so there's a bottleneck. Most of the overwhelming information gets to the V1 from the retina. There's another connection here through the superior colliculus and the pulvinar to the cortex, extrastriate cortex, but the overwhelming, more than 95% of the information goes to the LGN, V1, and to the other areas, higher order areas. Okay, so that's your connectivity sort of relationship. And one thing to remember, we'll get to that in a minute, you tend to think of this, the first thought is, okay, this is sort of a tree, this is the stem of the tree, and information flowing upwards, right? That's the way to think about this. But if you, from the anatomical point of view, you find that for each connection of an area, let's say from, I don't know, V2 to V3, there's a downward connection as well, okay? So there's just as much feedback from one area to the other, from the higher area to the lower area, as there's a feed-forward information flow from the lower area to the higher area. They don't go to the same levels in terms of the layers. Not the same layers, but there is still a feedback from each area back to the area feeding into it. Okay, so that is sort of the connection scheme, and it's easier to think about it. This way, and this was sort of general thought that was developed 20 or 30 years ago from lesion studies in monkeys. I'm not going to get into it because we don't have much time, but it's sort of, so here there's about 10 areas or so, and you're talking about two sort of information flow streams, one called the ventral one called the dorsal one, in which the ventral one is more devoted to object recognition, and the dorsal one more into sort of, I would say, vision into action, okay? And initially, people thought about this in terms of location. I think now it's more in terms of vision for action versus vision for object recognition. So that's sort of, and this was basically developed, this scheme was developed based on lesion studies made on macaque monkeys that showed that if you affected this area, you had, the monkeys had problems identifying objects, while if you affected the parietal cortex, they had more problems in terms of reaching or grasping objects, but they had no problem in terms of sort of recognizing the objects. So that's sort of the dogma in terms of sort of the division of labor between two streams, which you could see here. So you talk about this stream, the ventral one, and this one, the dorsal one. Okay, and again, if you simplify what you find in terms of functionality as you go from the retina to higher order areas, you find that first of all, there's a we mentioned about mapping, right? So this, that's not going to be, sorry about that. The mapping is a rule that you can find in virtually all areas, but it's very distinct and clear in the first retinotopic regions. But the other thing you can find also is that the stimulus, the optimal stimulus that drives the cells best, becomes more and more complicated, complex, as you move along this hierarchy. So initially in the retina, you've seen that it's basically dots that would fire the cells best. If you place the dot inside the receptor field, you get a robust response. As you move to V1, as you've heard, you know, work from Hubel and Wiesel, you go to edges, basically lines or bars or these gratings. And as you move on, you find that it's basically the stimuli that evoke the best response from the neurons, as much as we can tell, are becoming more complex. And, you know, the classic one is the face cell. Okay, so question number one, which we'll try to sort of allude to, we won't get much of a final answer, is how do you get this? What you have to do is generate sort of receptive fields, right, that are now sensitive to more complex stimuli, much more complex stimuli than initially. Now, I believe you did talk with Shaul about, you must have talked about Hubel and Wiesel and the fact, how do you build sort of an edge detector, okay, or a contour detector based on sort of summation of information from these sort of retinal ganglion cells or LGN cells, okay? But this becomes much more complex, as you will see. Another sort of basic feature that I need you to know is the fact that as you're moving from these lower order areas, V1, primary visual cortex, along this sort of the ventral stream, and it's also true about the dorsal stream, receptor fields become bigger. So if in V1, classical receptor field would be of the order of 1 degree, depending on eccentricity. As you move to V4, it would be maybe 4 or 5 degrees. And as you move to TE, to the infratemporal cortex, it could be 20 degrees, 40 degrees, much bigger. Okay. So sorry, I want to make it clear. I'm giving you some information, necessary information, but really what I want to deal with you is what are the problems, okay, more than just the information, okay? So problem number one that we would have to address is how do we get these very, very complex receptor fields? And problem number two, of course, is, okay, what's the neural code? I'm sure you addressed this in other issues as well, but okay, now you want to encode different faces or you want to encode different images of objects. How do you encode them? So this is something we will touch a bit here, but you will hear more about this in the lectures given by the students on Wednesday. Okay. So, okay, just a little little more details. Okay, so here's again the eye of the rhesus monkey, information flowing through the LGN to V1, and then now we're talking about the ventral stream, and you're seeing here 4 or 6 different regions from V1 to anterior IT. The anatomists, to make it more difficult, they use different names. So some would call this anterior IT, some would call this TO, the Japanese or something. So, okay, that's just to make things life harder for you. But the position of where this is does not change in terms of the brain. And in terms of, okay, so we see here now sort of this tree that I showed you here. This is the sort of the ventral branch of it. And what you can get from here is some idea of convergence and divergence in the cortex. So first thing to know, okay, as you move from retina to LGN to V1, what happens? If you talk about, Charles mentioned to you about the sort of the constraint of the optic tract, or not? So what is the constraint there? The constraint is, look at the number of sort of neurons that you have in terms of retinal ganglion cells representation. It's of the order of 1 million, okay? So the number of neurons that actually project their information to the brain is of the order of 1 million, okay? That goes through the LGN, again, 1 million. What you see here is sort of the latency. So you get, what I'm showing you here is sort of a feed forward, okay? There's a new image on your retina. You open your eyes, there's an image there, okay? And then, okay, so this million neurons here are active. And then, okay, things flow first upwards, right? And then we're going to talk about the feedback much later in my talk. But, okay, you get a basically upward information flow. And here you see how much time it gets before you get the first response in V1 and V2. So this is milliseconds, okay? So it doesn't get to the inferior temporal cortex, the anterior part of the inferior temporal cortex, before something like 100 milliseconds. So that's just the minimum time for information to flow to anterior IT. And I mentioned, okay, look here that there's sort of a bottleneck in terms of the information coming out from the optic tract into V1. And look here that now you're moving from 1 million neurons to something like 200 million neurons. So great convergence of information, okay? And then you can get an idea, okay, V2, another 150 million, and then there's some, did I say convergence? I meant divergence, sorry. But here you can see the convergence back into much smaller number of neurons. So you get an idea number in terms of the numbers and the times, just so you have a sort of a picture of things. And here's sort of the complementary picture here. And that is, okay, what are the best stimuli as you move from V1 to V2, V4? So this is along the ventral hierarchy. And notice TO here and TE instead of posterior and anterior IT, just to make things more difficult. But again, you look at V1, you see, okay, these neurons which are tuned to specific orientation or to gradings of a specific spatial frequency. And then as you move to V2, okay, look at the times. Okay, here's the size of receptive fields. So the receptive fields become bigger. By the way, you see that there's a great diversity. Some are very small, some are bigger. But the diversity is such that at the center of gaze, the fovea, the receptive fields are very small. As you go to the periphery, the receptive fields have become much bigger. So that's true for all levels here. So you see some of this diversity has to do with center and periphery. And you can get an idea what is the best stimulus as you're moving from V1 to V2 to V4 and so on and so forth. And here's what you get in anterior IT. Suddenly you get neurons which are, would be responsive most to this flower or to this vase or to the tiger here or to the face of the monkey. Again, we'll get to that. How do you, we won't give a good answer, I think, but how do you generate these receptor fields from the building blocks of earlier visual areas? I don't, you know, you look at it, it doesn't seem very to make much sense, okay? When you hear more about it, it wouldn't make more sense later, but this is something that we have to, I think this is a sort of caveat in our understanding, which it's time to fill in terms, and I think it's time to fill through theory, okay? What should we look for in terms of the right alphabet? But last you get here a picture of the activity that you would find. Okay, so this is the monkey brain. Am I speaking too fast? People always say that. So am I giving you too much information? I need to hear from you to see that, you know, you're following me and everything's fine. Okay, so what you see here is, again, just to give you some schematic of the sort of development of activation. Okay, so before 40 milliseconds, nothing gets to the cortex. Okay, first activation is found in V1. MT is the motion sensitive area. And then you see, okay, this is sort of a time flow. Later you see activation in these higher areas and in frontal eye fields. And this is sort of gives you a picture. I'm not going to go over the whole detail, but gives you sort of a picture of, okay, information reaches first the occipital lobe, and then travels to the frontal areas, and then there's basically, by 100 milliseconds, there's the feedback loops start operating. Not just within an area, that's immediate, but it's also within, between areas. That is, from higher order areas, feedback back to lower.
What's the difference between the yellow and red colors?
Gee, I don't know, but I think it would be. It could be. I mean, let's see.
Could be adding of new.
Yeah, maybe it's the new ones. Yeah. I think all the colored ones are active. That's for sure. But it could be the. Check it out. I would say that it's probably the new ones. I mean, see here. This is. So yeah, the old ones, which were already active before, are colored in orange. Okay. How am I doing for time? Not very good. Okay, so here again, I just want to give you some flavor of this. So what would you record if you're recording from V4 or IT? So notice here. First, what you can see in these circles are the receptor fields of individual neurons. So what you find is, first of all, you see that the receptor fields grow. That's one thing we talked about as we go from V4 to IT. So V4, remember what V4 and IT is? You don't remember, of course not. Here's V4, and this is IT, okay? So V4 is like two orders away from V1, but still the input to the higher order area. Okay, so receptor fields become bigger. Note another fact which is important. You see left hemisphere and right hemisphere. So I didn't mention this, but this is something which is kept very strictly in early visual cortex. That is that the right hemisphere encodes the left visual field with respect to your fixation. The left hemisphere encodes the right visual field. And you see that there's not much overlap. You see this is the vertical meridian. Right hemisphere, just left visual field. Left hemisphere, just right visual field. As you're going further up, suddenly the receptor fields become bigger and they invade into the ipsilateral side. So they include both sides of the visual field. And what you see here is just an example of sort of the response of a single neuron as a function of time. So at time 0, you turn on the stimulus. You see here the latency we talked about. It takes about 100 milliseconds before the cell starts to respond. And this is the gray period here is a time in which the stimulus was on. And you see that there's sort of a transient response, then a drop of the response. And as you close the picture, you turn off the picture, there's some residual response which dies out within another 100 milliseconds or so. And what you see here is just to give you an idea, that's one neuron recorded from IT, and these are sort of, you rank order the images that the investigators used. Not much clear. Okay, is it a chair cell? I don't know. Suddenly, what's this? Matchanat basar. I don't know how to translate that, but you wouldn't want to put your hand inside one. That's for sure. And here's the worst images. Again, it's unclear here, and here's just another example. And you're going to hear a little more about this in the near future. I'm moving into just to give you an idea. Okay, but one classical example was these face neurons that Charlie Gross found in early 1970s, and they seem to respond to monkey faces, but they also sometimes responded well also to human faces, also to some smiley caricature of a face. And here's just the example showing that, okay, a face in itself elicited greater activation than the full human body, and without the face, there's not much of a response. So these seem to be face cells. You'll hear about them soon in much more detail. So here we are. Here's a later study in which they tried to look at this a little more carefully. So they presented, I don't know how many images are here. I'd say of the order of 100, 150 images, okay, 120. And what they do here is sort of they rank order. So each one is flashed for a very short period of time, and now they look at the response to each of these images. And the question is, okay, what's the best stimulus and what's the worst stimulus? So you can find that, okay, if you rank order them, they do indeed seem to be face cells in the sense of okay, look at this one, and okay, so this is number 1, number 10, number 11, number 20. They're all faces, right? Now, there may be another face here, so there's some false negatives, or this one here, okay? But in general, and so it's from a different perspective than this one, of course, profile, also this one. But in general, they if you take this 150 or 120 images, they do seem to be, this neuron seems to be, have clear preference for faces over others. Okay, so the first thing is this issue of, okay, is this something that's prominent to, what do we make of this, okay? So this is a more elaborate study done by Doris Tsao about 10 years ago, in which, and I bring this for two reasons. The first reason is that it shows that, well, nowadays I'm working with functional MRI. Now, a lot of people say functional MRI. What are you studying in functional MRI? The answer is we don't know. But the answer is we don't know, okay? We are studying something which is sort of a correlate of neuronal behavior, but is it a reasonable correlate of neuronal behavior? This is where this comes handy. Okay, so this is the work done by Doris Tsao, in which what they've done initially is they sort of put the monkey inside the scanner and presented images of faces. And now what you find here is these blobs, these famous blobs that you find in MRI. And this is sort of functional MRI here, okay? So you find that there's these blobs of activation that are voxels, which are more selective to faces over other objects, such as bodies, fruits, gadgets, or whatever. Okay, so face-selective voxels. Okay, great. Is there any meaning to this? Well, the next thing, this is the advantage of doing monkey electrophysiology. So what you see here is they've moved an electrode into this area. By the way, you can see the damage caused by electrodes. You see this? The black area here or here. These are the individual tracks. If you do this enough times, you get damage like a Swiss cheese from cortex. So don't do this at home, okay, to yourself at home. Sometimes it is used. You've heard lectures by Fried. You've heard, so it is used in rare cases in humans when to treat epilepsy and to find where the focal area of epilepsy is, but it's not used in other cases, of course.
Is it not possible to reach it from the side and avoid this long?
It is, but there's other problems with that. Okay, mainly it's not. It's a tricky issue. It has been done. Yeah. What you can see here, the reason I brought this is to show you that, okay, now you focus in these areas, and now you record from neurons with an electrode in these voxels, which seem to be phase sensitive on the level of fMRI, and this is what you get. So, okay, what do we see here? You see here, this is the y-axis here is cell number, and the red dots here correspond to high activity for a specific image. it's organized such that the first 16 images are images of faces. The next 16 images are bodies, fruits, gadgets, hands, and so on. And what you can see for yourself is that, okay, this is monkey 1 and monkey 2. The vast majority, more than 90% of the cells, are selective to faces over other images. It's pretty overwhelming. I think it's quite impressive. Look at the second monkey. Now, there's some neurons, a minority, which are not face selective, but overall, they are face selective. And to me, this was one of, there were other works done in primary visual cortex replicating what we know about retinopathy, but this is very strong proof that, functional MRI is not a useless imaging technique in terms of understanding basically neurophysiology, because here you are. Of course, what you're seeing is the grand average of these neurons, and therefore it's selective to faces over other objects. Okay, so clearly what you can see here is that there are specific clusters that are face selective. Now that suggests something, that maybe we have some domain specificity. Again, do we take a break? I see that you've been looking at do we take a break here? Usually we do. Okay, so I have 3 more minutes and we take a break. Okay? And I was talking about the issue of domain specificity. And domain specificity means that, okay, there's a specific domain responsible for face recognition. The other option, of course, that it's all distributed, and you'll hear something about this from the students and from me after the break. I just want to add up one piece of information before we take a break, and that is work that has been done by Hussein Asteki from Iran, by the way, from Tehran, about 10 years ago. I think a very nice piece of work. And the idea here was, okay, let's tune in on these clusters, and now not just record from the neurons, but try to activate them as we're giving visual stimuli. Okay? Now, they weren't the first. The first ones were people who have done this in area MT, Bill Newsom and his group, in MT in the early 90s. But it's a very, I think, useful technique to study the issue of causality. What if you now activate these cells? Can you influence the monkey so that he would judge an image to be more face-like when the neurons are activated compared to when these neurons are not electrically activated? So the idea is the following. Okay, you're presenting images, these sort of degraded images, and they could be more face-like or more not face-like, like this flower-like, okay? And you can play the level of noise. So this is a completely degraded image, and this is 20% degradation. This is also 20% degradation. And, of course, I made a mistake here. This would be 80% signal, and this is minus 80%, or 80% signal again. So this sort of, this is clearly distinct as a flower, and this you can still recognize as, I guess, כדור פורח, no? Whatever, yeah, a ball maybe, I'm not sure what it, yeah. Okay, and not really critical. What you do is the following: you present the image for a short period of time, and then you microstimulate. So again, so what did we do? Let me explain. Okay. What did we do? We moved the electrode. We recognize that there's here a sort of a face cluster. So we move the electrode, place the electrode at the center of a face cluster, okay? And now we basically stimulate through the electrode, okay? So what he does is give...
Yeah, we can.
I'll draw and then explain. Okay, so what do? You give, these are pulses that you give through the electrode, electrical pulses. Okay, 1, 2, 3, 4, 5, okay? And there you could increase the frequency or the amplitude of these pulses, but these pulses effectively drive the neurons, okay? So now you have a condition in which not only does the visual stimulus drive the neurons, but the pulses through the electrode, drive the neurons, and basically the neurons fire on a one-to-one basis to each pulse. Okay, unless it's a very, very rapid pulse in terms of frequency. So what you're basically doing is mechanically driving these neurons. Now, these are neurons which are phase selective. So what would be your hypothesis if you have this technique at hand? What can you do now? And I just described to you, okay, the images. So let me just help you. Okay. No, you answer. Okay. Okay, so you have these. Let's think about the logic here. Okay, the logic here is to establish causality. Okay, I want to activate these neurons and show that if I activate these neurons, I can make the monkey choose an image which is sort of completely noisy, or even an image which is not face-like. I would bias the monkey by activating these face neurons to say about an image that it's face rather than non-face. Okay? So you want to give me the answer, or shall I go to the next? I'll go to the next due to lack of time here. So here's the result. What you do here is you plot on the x-axis the visual signal. 0 means a completely random image. Plus means it's a face, and minus means it's non-face. Let's say a flower, okay? And the y-axis here describes face response. So the monkey, you're registering here the behavior. The monkey, on each trial, he's getting these images, previous one, an image like that, and at the same time, there's a stimulation, electrical stimulation, which could be together with the image or a little delayed compared to the image. And now the electrical stimulation activates these face neurons. And now, what would be the effect? So, okay, without electrical stimulation, you'd expect that at a visual signal of 0, completely random, the monkey should choose, the, say that the image was a face at 50% of the times. Let's think, okay, again, so this is face responses. It's not correct responses. It's how many times did the monkey say that this was a face in the image, okay? Let's think about, the cases, the extreme cases. So let's go to this. This means it was a very clear-cut face, okay? 50%, it had a visual signal of 50%. What do you expect the monkey to do without electrical stimulation? Say that it was always a face, right? So it would say it was always a face, or it should reach 100% here. It's almost at 100%. Let's take the other extreme. Let's say that the visual signal was non-face-like, very extreme non-face-like. Then you expect the monkey to say, okay, it's not a face. It's a non-face. So the percentage of times he would respond telling you that it's a face should go down to 0, right? So, and at 0%, we expected this to be maybe at 50%. So it's sort of a sigmoidal function, which we've described its extremes. Now let's think about the stimulation. If it has an effect, what should you see? You should see that, first of all, let's take the non-stimulated case, there should be an upward. Think about the case of 0%. At the case of 0%, there's nothing in the image. But with stimulation, the monkey tends to choose to say that it's a face he's more likely to say that than without stimulation. Okay, now there's some subtleties here in terms of when is the timing of the electrical stimulation. But what you see, or you could look at it a different way, the curves shift leftwards, meaning that now the monkey has a clear bias in saying that the stimulus that has been shown is more face-like than without stimulation. Okay, it's not a huge effect, and it is very much dependent on position. That's what you see here on the inset. But basically, what you've shown here is that by electrically stimulating this cluster of neurons, you can affect the monkey's perception, or at least decision, to say that it's more likely to be a face. I think we'll take a break now, and we'll talk about this later. Yalla, הפסקה. לא הבנתי, אוקיי.
לא משנה, אני עושה לגרות אותי, כן.
אוקיי, אתה תשתמש באוטו גנטיקה פיזית.
או חשמלי.
אוקיי.
אוקיי?
כן.
וכאילו המטרה שלי באמת זה להפעיל את התאים.
כן.
אבל גם אם אני בסוף מקבלת אפקט הפוך, אני יכולה להגיד, אוקיי, אבל אני מפעיל תאים, אבל הפרעתי לפעילות טבעי שלהם. זאת אומרת, יצרתי כאילו...
אולי תאים איניביטוריים, שמדכאים את ה...
או לא יודע, זה לא תמיד סינכרוני בתדירות מסוימת ו...
כן, באוטוגנטיקה יש יתרון גדול ביחס לשיטות האלה, שאתה יודע את מה אתה מגרד.
נכון.
אז אתה יודע להגיד אם אתה מפעיל תאים איניביטוריים, אתה יודע, אתה עושה טרגטינג של תאים פירמידליים,
אבל אפילו מעבר לאיזה תאים, איך אתה מגרש? איך אתה, כאילו, על הפעילות
איך אתה מגרד? אתה יודע, כאילו, כמו שאמרתי, שאתה נותן פולסים כאלה שמגיבים, קצת, אם זה לא בתאר זה מטורף, אחד לאחד על הפולס. אתה יכול לרשום, נגיד כמה, אתה יכול לרשום במקביל.
מאוד שונה מהפעילות הטבעית שלנו.
זה נכון, זה נכון.
זה לא מפתיע כאילו שזה גורם להעצמת ה...
לא, זה... בוא נגיד ככה, זה לא היה ברור שזה יצליח. כי את צודקת, זה עוד פעם, אנחנו מגיעים מכיוון אחר, עוד פעם היה איזה של ניורל קודינג, כי את צודקת לחלוטין. זה לא, כי מה שיוצרת פה זה בעצם איזו פעילות בו זמנית, שהיא מסונכרנת של כל התאים.
כאילו אי אפשר להגיד שאני עכשיו מגבירה את הפעילות טבעית. אני יוצרת פעילות.
לא, את מגבירה, לא, פה אני נותן גם גירוי ריאתי. אז זה בצמידות לגירוי ריאתי, ולכן זה כן להגביר את הפעילות הטבעית. זה נכון, עוד דבר, לא, זה נכון עוד דבר אחד, כאילו אפרופו.
הבעיה שכל תוצאה אתה יכול לפרש באופן שאתה רוצה.
כן, אבל אי אפשר להתווכח עם תוצאה כזאת. אם זה יוצא הפוך, אז הייתי יכולה להגיד, זה לא...
למה אתה יכול להתווכח? אפשר להגיד, טוב, אז אולי...
לא, היה לך פה הנחת האפס זה שאתה תשפיע על ההחלטה של הקוף, כך שתהיה העדפה לכיוון של הפייס. אם אתה בתוצאה הפוכה, היית בבעיה, אני חושב. ורוב מוחלט של התוצאות הן כאלה.
למה, לא הייתי יכולה להגיד, כאילו, שיניתי את הפעילות...
הפרעתי לפעילות הטבעית על זה שהכנסתי גירוי חשמלי לא טבעי להם, אז הגיוני שזה ירד להם.
דפוס פעילות דינמית.
הייתי יכולה להצדיק גם את התוצאה.
תראה, בוא נגיד, אתם צודקים דבר אחד, תשמע, זה מאוד קרוב, זה מאוד מאוד גס. אתם לוקחים פה, נותנים פטיש 5 קילו על התאים, גורמים להם ירי מטורף, והנה אוקיי, הראיתם שהשפעתם משהו על הקוף, אוקיי? עכשיו בוא נגיד ככה, אם היית יוצר ליז'ן, היית מצפה שהתופעה תהיה הפוכה.
כן.
נכון? צודק, שרק תעלה את הקצבים למשהו מטורף, אתה גורם להשתקה אחר כך של הפעילות של התאים, אולי אתה מצפה למשהו הפוך, אוקיי? אבל באופן עקרוני, הנחת האפשר החולים הייתה שתקבל אפקט בכיוון הרצוי ולא בכיוון ההפוך. ועם אופטוגנטיקה יש את העניין, באמת שזה הרבה יותר מתוחכם מזה שיש לך יכול לעשות טרגטינג ספציפי. ולא רק זה, אתה יכול גם להתחיל לעבוד עם איזשהו קידוד יותר מסובך. פה זה רייט קוד, אנחנו נותנים אותם בתדר של 50 הרץ, 100 הרץ, 200 הרץ, וכו'. מה? כן, יש לך מכשיר שהוא הרבה יותר עדיף. הלו. טוב, עכשיו אני צריך רק איזה מים ואנחנו נראה.
ורואים מה
חגי, אני לא גנבתי לך יותר מדי מה זה.
יש לך 2 שקופיות.
זה דווקא טוב, כי אני רוצה שתתרכז.
לא, תוכל לעבוד את זה כבר.
ראיתם? כל הרב שאני מראה, אבל זה לא שאתה יודע, אני רוצה לחסוך לך את הרקע.
כן, כן. אני אמשיך כאילו...
לא, אתה בכל...
זה רק, 2 שקופיות האלה, מה זה
אינטרודקשן
שלי?
מה?
כן, אני לא יודע מה יהיה בשעה הזאת.
אני ראיתי שאתה במתח.
לא, יש לך 2 שקופיות שהן באינטרודקשן שלי ואני חייב להציג אותם.
הוא בכל זאת, לא קרה כלום.
ואז אתה יכול לרוץ עליהם
כן, לא קרה כלום.
רציתי לשאול למה אנחנו לעשות
לא טוב.
זהו, מה? למה?
כי הם לא חיות רועות, תתחיל לזה. הם חיות ליליות.
תמיד כל המחקרים האלה זה ממתק ומעלה, כאילו מקופים ומעלה.
לא, א' מנסים לעשות את זה, כי יש לחץ מאוד גדול. עשינו עבודות היום, כאילו שבאמת מוכוונות עכברים וחולדות, גם בגלל שיש את הכלים הגנטיים שהתפתחו בצורה האופטוגנטיקה, אתה יודע, וואו, אתה לא יכול לעשות את הדברים האלה, אופטוגנטיקה מוצלחת עדיין היום בקופים. אז דבר שני, יש את העניין שהלחץ של, אתה יודע, animal rights movement, שכאילו רק ידע, אוקיי, אבל העניין הוא...
אבל עכברים אין IT, אין זה, כאילו, אפילו לעורב הוא עושה כלים, העורב יכול לקחת זה ולעשות לו כאילו הוא מזהה חפצים, יש לי חפצים אצל...
בוא נגיד ככה, המרחק הוא, מפה לאדם הוא הרבה יותר קרוב מאשר מהעכבר לאדם. עכשיו, מה המטרה שלך? זה באיזשהו מקום, אתה יודע. בוא נגיד ככה, לגמרי ברור שאם לא היה
קודם
שיקול
הייתי
הערכי,
חושב
אם לא היה לך את השיקול האתי, כמובן שברור שיותר נכון לעבוד עם הקופים. אתה לא עובד עם בני אדם, אבל אתה רוצה, אוקיי. עכשיו אתה אומר, אוקיי, רק אני לא רוצה לעבוד עם קופים מהסיבות האתיות, נכון? כאילו זה ברור שזה השיקול.
אולי גם אתה רוצה להיות מערכות פשוטות יותר?
זה נכון, אבל אם אתה רוצה להבין את האדם, אז אתה רוצה, אתה צודק, זאת אומרת, צריך לא לקחת זמן, זה הכל. you want me to turn it off? Is this?
No, it's just for testing a microphone.
I'm
Low
sorry.
end
Yeah.
will kill it.
Okay. So last thing we've done was talk about these electrical microstimulation effects. By the way, during the break, someone asked me, this can be done now, not in monkeys though, but this can be done now much more effectively using optogenetics, which I'm sure you've heard about. And the great advantage is that, you know, with electrical stimulation, you're stimulating all the neurons near the electrode. Of course, as you're increasing the amplitude, you're going to recruit more and more neurons, but you're recruiting interneurons, which could be inhibitory and pyramidal cells and everything. Of course, with optogenetics, you can now target a specific class of neurons, and you may be able to learn something beyond this, okay? It is somewhat sensitive to the timing as well. So if you've seen here, this is just one experiment, but it shows you that, you know, when do you get the greatest effect? You get actually the greatest effect when the electrical stimulation is somewhat delayed with respect to the time of the visual stimulation. You have any idea why?
Makes sense because it was showed us
Takes time to get to.
Exactly, because it takes something like 100 milliseconds to get for the signals, the visual, basically, to get the activity from the neurons. And this is what's shown here. That's the multi-unit response. You see here when you're showing faces, here's the response. So the response peaks at about 150 milliseconds. So if you're giving the electrical stimulation here at time 0 to 50, this is not the time at which visual stimulus has reached. So it's sort of the wrong time, okay? I mean, think about it. How do you interpret these signals? What you do is you basically read out the signals from these phase clusters, and if there's activation or activity in these cells at the wrong time, presumably there could be a gate saying, okay, this is irrelevant. Although, as you see here, there's some effect also when it's in the wrong time. It's greater when it's in the right time. Excuse me?
Can you repeat, please, what was the difference between the red and blue?
Yeah. Blue is non-stimulated, so this is just a basic performance, okay, as a function of the visual signal, okay? The red is when it's the electrical stimulation is delayed by 50 milliseconds with respect to the onset of the visual stimulus, okay? And then you get a greater effect.
But then you
Oh, and here, I didn't mention that. I only said that this is the time it takes for so what you see here on the y-axis is the multi-unit response. So you're recording from the electrode at times when you're not stimulating. And you see here, this is for faces and this is for non-faces. So you see that this is a differential response to faces. Okay. Okay, now so here, we were talking about a sort of a phase cluster. What about other categories? So here, when you record from the monkey, just to give you an idea that it's not very clear what makes these cells fire, right? So look here. I mean, here's a cup of tea from Queen Victoria, which generates very nice activity, but this one, for some reason, the cell doesn't like it all, or this one. But it does respond to the cow here, or to the bear. Excuse me? It's the same cell, yeah. So you see here, these are different stimuli used, and this is so just to give you an idea that, okay, the cell responds to this cup, but does not respond to that cup. It doesn't respond to the duck, but it does respond to the rabbit, okay? What do you make of it? Unclear. And this is just the time 0 is the time when the stimulus was on. So you see, this is sort of the histogram of the response. Okay, so it's unclear. Could be. Any ideas? Yeah, one option is that, okay, the receptor field properties now are such that it's not specific to a specific object. We would think about how to interpret this in terms of sort of a coding scheme. But another option, a more trivial option is some feature which is common to all of these, which we cannot detect by eye just by looking at this. I don't know if there's maybe something here, So that actually the receptor field is very small, and there's sort of a contour here, which is, you know, common to all of these. I don't think that's the case here, but they've checked this, so that's not the case here. So I think it's more complex in the issue. It could be, of course, in a different sort of, could be a feature in a specific spatial frequency. So if you Fourier transform this, look at a spatial frequency, maybe it's, you know, something oriented in specific spatial frequency. But it doesn't seem to be the case. So I mean, this is difficult to interpret. First of all, how do you get that sort of tuning? Doesn't seem to be tuned to something clear like a bar in V1, okay? And second, what do you make of it? Okay, how do you use this? Because, again, the thought was, following Hubel Weasel in, I think, first 20 or 30 years after that, to think about this in terms of sort of a hierarchy in which you get, you know, as you move up, you get to neurons which are sensitive to more and more elaborate sort of images, but they remain sort of category specific. So you have face cells or maybe house cells or whatever. Of course, it's absurd to think about this when you're trying to encode all the sort of visual objects that you may encounter in your life, or that's what it seems to me at least. But what is the alternative? So what would be the alternative? Yeah.
For example, that the whole population of these IT neurons together could, for different objects.
Exactly. So, of course, I'm sure I'm not the first one, and not the last one you'd hear this. Okay, so sort of a distributed coding, in which you can think about of each neuron sort of. The analogy here is, of course, from the computer, in which each neuron is a bit, which is either on or off, if you want, okay? And it could be on and off for different objects, and there's no coding of a specific object in the sort of the firing of this specific neuron, but it's across the population in which the encoding is done. So yeah, I think that's one way to look at it. And okay, but I want to go to something, again, to single neurons here before we go into this encoding issue, which will be dealt with in the students' talks, and that is something that Keiji Tanaka did in the 90s, in which he sort of tried to see if you could look at the images which evoke a response from the cells and try to see if this is to reduce the image in some way so that you would get the basic image which still elicits a strong response from the cell. So what he did was the following. I mean, this was Japanese in the 90s, sort of. So he brought the toys that he from his kids' room. Okay, so you recognize what this is? Huh? It's from up. It's a tiger, okay, from upward. Okay, so you see here, I guess, well, I'm not sure. That's what I guess it is. You know. Okay, it's a doll of a tiger or something like that. Okay, and then so what he did is take this now, sort of reduce it in some ways, make it simplify this, and sort of instead of this image, use this image. And if you still get a robust response to that, change that in the following way. Get rid of the sort of the oval shape here, make it a square, and so on and so forth. And as long as you get the response from the cell, go on in this reduction, sort of stimulus reduction method, until you get, once you get to, you move from here to here, you don't get much of a response, or here you don't get much of a response. You say, okay, this is the basic stimulus which elicits a response from this specific cell. And this was in posterior IT, not in anterior IT, in the area. And here's another example, just to give you an idea. Okay, so what he did, use objects. Then I failed to mention, this was Japan in the 90s. What did the Japanese have in the 90s that the rest of the world didn't have? Digital cameras, okay? So he had the option of taking a picture of these objects. Okay, so first, he sort of flashed the object, sort of used the object, showed that the cell responded to it. Then he simplified it in some ways. Okay, now sort of making a silhouette of this. Still got a response. Moving to this lollipop, right? Arctic, okay, so, no surprise that lollipop got the best response from the cell. And then as he moved, no lollipop, no response whatsoever. Okay, so you get an idea that this sort of a simplification method. What do you think of this? Does that make much sense to you? Well, how do you react to that?
No, no, okay, he doesn't assign meaning to it. What he's trying to do is sort of a method for simplification of the stimulus to find what are sort of, what's the logic of it, to find what's the alphabet, okay? So sort of in parallel to language, you're saying, okay, in language, okay, not in Chinese or I don't know about Japanese, but in sort of our language, you can build basically virtually endless numbers of words from the sort of building blocks, which are just letters of the alphabet. Okay, so maybe let's think about sort of find what are the sort of letters of the alphabet, the visual letters of the alphabet, from which we could bring, okay, so it's basically building block of basic features from which we can sort of now build any object we want from sort of a cluster of these sort of alphabets. That this is the same as that. Okay, okay. I agree. I think you want to say something before I'll. No, you go ahead. The idea is for you to speak. Okay.
I think it makes sense, but there are a lot of different ways to make a reduction of one object.
That's exactly what I want to say. Okay, so basically it seems somewhat arbitrary. Okay, it makes sense, but it's arbitrary because I mean the question is, what if you initially started by doing something else? I mean, you could think of very, as you said, a very different way. It's not necessarily logically necessary to go from here to here. Okay, clearly. Okay. You may sort of emphasize the brightness here or something, okay, or the curvature here, other aspects, and then you might end up in a completely different sort of, you're taking a different path along the tree, and who knows if you're going to get to this one. You may get to someplace else, okay? Because I don't think, of course, the problem is, of course, that we don't know what is the optimal stimulus for the cell to start with, right? So having reached this, okay, so they're saying, okay, look, here, this is even a stronger response than that one. Okay, that's another point that I failed to mention. Okay, but I'm not convinced. Okay, I think it's a nice idea. Okay, but I'm not convinced that it has any sort of theoretical merit. That's the problem. Yeah, just one more aspect to that, and then we'll take some questions. So these are the features that he found. Okay, using this technique. Okay, these are the features that he found. Are these sort of basics of visual alphabet? Good question. It's not what I would guess, okay? I would think maybe T's, L's. I mean, there's some, this is something, again, if you were to talk to people from computer science, they'll tell you that T's are important. But the others, you know, the lollipop effect, I'm not sure. Fingers, I don't know, really. It's a good question, but I'm not really convinced about this. However, time for questions, sorry.
Because if you follow the analogy with language, you can break language apart first into, you know, the alphabet and phonemes, but there's also the smallest units that still carry meaning, morphemes. There's a big difference between those two. So could you perhaps try break down instead of just into random bits of pictures into somehow meaningful bits?
Okay, so first I think the question is, the issue here is theory, okay? And the theory is, okay, what should you look for to start with, okay? And this is where I think it's problematic. We were talking about, first, there's two issues. First of all, it seems somewhat arbitrary. And second, I think it's related. It's the fact, okay, what should you expect to find? If you have a sort of a driving hypothesis, you may end up with a sort of actually find the right elements. However, the surprising thing is that when Keiji Tanaka actually did this, and he's done this for years, he found that, okay, here's sort of he found columns. So you've heard about columns in V1. So surprise, surprise, you find similar columns in infratemporal cortex. And if you're sort of, so this is sort of cortex, and if you're sort of going inside, you find, okay, so think about, okay, I was describing before a case in which Here's the cortex. You move an electrode in, and you record from the neurons here as you're sort of moving, say, normal to the surface. Okay, layer 1 to layer 6 here. And what you find is that the neurons have similar features. So if you're doing this here, you find that they're sort of responsive to this oval-like, egg-like shape. And if you're moving to here or there, maybe it would be sensitive to the direction of illumination. You do this in a different position, and they seem to be, they're not organized in a random, the sort of the preferred features are, again, sort of in sort of clusters. So here's another one in which it's sort of ellipses going to circles. And here's the face clusters that we were talking about before. So, and furthermore, I may not go into this in depth, but you can use a different. So this was using as an electrode, a more advanced sort of technique that was sort of developed in the mid-90s was optical images. Have you heard about optical imaging? So optical imaging is basically optics. You don't record from a cell by cell, but you sort of you open sort of a window in which you can sort of basically picture the cortex. Typically, and what you have is either voltage-sensitive dyes, so you're sensitive to basically the electrical activity of cortex in a window of maybe 1 to 1 centimeters. And basically, the voltage-sensitive dyes are such that they allow conversion from the voltage of the neurons, so the potential of the neurons, into a sort of a code which can be sort of a code in terms of brightness, which can be imaged. And when you do this, what you see here is, okay, in green or in blue or these sort of areas are areas which are columns sensitive to these specific sort of letters of the alphabet that Tanaka found. So here's our lollipop neurons, and they're sort of clustered, and you can see them in using an optical image technique. So, and what you see here, just these are the blood veins. So they allow you basically to sort of register where they are in terms of the cortex. And this is sort of 1 millimeter. So you see here a window of maybe a centimeter by centimeter. And with optical imaging, you do not record spiking. You record basically just, as I said, electrical activity, which is also synaptic. But you get a nice confirmation what Tanaka found with single unit recordings. And finally, here's just an example of sort of phase clusters. So you see for sort of from the profile and straightforward, and you see that these phase clusters are organized. You see them here, so that as you're moving from one, you're sort of rotating these faces, you get some shift of these clusters. So there's quite a few of them. It's not just one, 1, 2, 3, 4, and here's 3. Some of them are lost here, but if you see here, you can see that there's clusters here which are sensitive also to the direction of the face. So maybe faces are indeed something, a domain which is unique in some ways, but it also brings up this issue of, okay, this alphabet that Tanaka reported. He does have confirmation using a completely different technique. Okay, and I want to say a few words. There were some questions? Yes, please.
I just wanted to ask about the neurons described a few slides before that.
Yeah.
Did he make sure that these neurons are not modulated by memory or context? So did he put the monkeys in?
No, typically, okay, so typically, the monkeys are anesthetized in these experiments.
Even in the IT experiment?
Yes. So, and this is posterior IT. Remember we talked about, so this is, posterior is back, so it's sort of not the final. I mean, memory-related activity is typically found in anterior IT, okay? And this is, as I said, in anesthetized animals, okay? And if you're looking for the alphabet, probably you want to go to sort of the more posterior regions. Okay, and I want to say something about functional MRI in humans. Okay, what's that got to do with humans? Well, first of all, we know from studies in humans that this is an old slide, but you can find the same sort of corresponding areas to V1, V2, and so on and so forth. And you can see here what's the best sort of stimuli for these different regions. So here's just sort of a view from the midline. Here, this would be primary visual cortex V1, and this is V2, and so on and so forth, dorsal and ventral, and light coming from retina, LGN going to V1, and then spreading to the different areas. And the reason I'm showing you this is that a lot of what I've described actually has been corroborated or found in humans as well using functional MRI. So I mentioned Doris Tsao doing the work in monkeys. Well, here's the region. This is work that has been done by Rafi Malach 15 years ago, in which he found that there's these areas, again, a little more anterior. This would be here. The occipital pole would be primary visual cortex, so a little anterior and more on the ventral side, left hemisphere, right hemisphere. This is the ventral view from below, left hemisphere and right hemisphere, that are active to these objects. And they could be either a face or a cat, but they could be also these Henry Moore sort of statues, which we recognize as objects, although they are new to us in terms of their specific shape. And these areas are more active during when subjects see these objects compared to textures or to sort of random textures. And later, Rafi and Kalnick Spector have been able to show that these areas are active. Remember I talked about the fact that you can extract an object, this issue of constancy. Another way to think about it is that you can extract an object from different cues. It could be from luminance, that's like classic, but it could be also from a texture difference. So here's the bird in which textures, if you were to take the mean luminance here and here, it would be the same, but the texture is different. So in terms of spatial frequency content, it will be different. And this could be also, you can extract the object from motion. So this isn't, it's not shown here, but if you think about, there's no contour here, but the dots within this region move coherently against the background. And then immediately you would recognize that there's a dog here just from the motion. So each of these cues activates these regions, suggesting that they are sort of object selective irrespective of the cues that you use.
Do they activate different areas or the same?
So so what you see here, I mean, there's some correspondence or some dissimilarities between these different cues. So I'm just giving you the general view here. We could.
They're claiming that there are different areas.
I think they would claim here. They would claim here that these object selective areas are activated by either of these cues, okay? And disparity, and there's other cues which I'm not getting into. And another classical example you've probably heard about is this FFA that Nancy Kanwisher and colleagues have found. FFA is the fusiform face area, which is active when you, in humans, when you show faces. And there's another area which she called PPA, parahippocampal place area, which is activated by views of scenes, outdoor scenes. Okay, and this is just to give you an idea of how does a simple fMRI experiment goes. Sort of block design, which you sort of change from a time where you show fixation. So here's the time scale. Okay, we're talking about a signal which is of the order of seconds. So this is. Each time point here is 2 seconds. You sample the activity at 1 or 2 seconds. Okay, so you're not talking about milliseconds. You're talking about seconds here. And in terms of spatial resolution, you're talking about something like a million. Just to get an idea, a million. The average activity of a million cells that you're averaging. And basically, you see that you get this robust activation. In this FFA, whenever there's faces, and it drops down, there's not much activation when you're showing houses and so on and so forth. And you get an idea, by the way, of the variance or the noise in the technique. Where do you see the noise, by the way? Sorry to keep you busy, but I want to see that you understand. So, okay, First time, second time you show faces, third time, fourth time, fifth time. You get an idea, okay? So this isn't quite the same, right? That's the noise, okay? So that gives you some idea about, and here's houses, so it's not exactly the same. Although you're averaging, this is the average activity of the whole region of interest, so it wouldn't be 1 voxel. It's actually, I don't know, maybe 50 voxels or so. You'll still get quite a degree of noise here, and most of it is not biological noise. It's noise just due to the hardware using, so basically scanner noise. So that means you need a lot of averaging, right? You need a lot of averaging to see any statistically different situation comparison, say, between faces and houses. Yes?
This is one subject only and one block only, right?
Yes, it is one subject and not one block. 1 block, 2 blocks, 5 blocks. But one side, yeah, I wanted to give you some idea, okay, but what does the noise look like?
So it's much better than average fMRI experiments. So this is one of the most noise-free signals you can ever get in fMRI, right?
Let's not exaggerate. It's one of the better subjects for sure. Okay. Yes, it's a noisy measurement, but that doesn't mean. I mean, that does not mean that it's worthless. Okay, it's a noise. No. Yeah, it is noisy. It is noisy. You have to average a lot. Okay. Next. Okay. One point about face blindness. Have you ever heard about prosopagnosia? Yeah. So the ones who studied psychology, I'm sure, did. That's just a demo. Okay, a prosopagnostic person. I mean, why am I mentioning this? Because, okay, we found that there's a face clusters, that are multiple face clusters, both in the monkey and in the human. And the question is, okay, is this really, and we found that if you actually stimulate a face cluster, you can affect behavior in the monkey, okay? Now the question is there any sort of relationship to any, why am I talking? You're all looking, okay, how many faces did you find? Okay, so the point I want to say is that prosopagnostic, looking at this, would see the vegetables, but it would not see any face here, which I believe, if anyone doesn't see the face here, please let me know. Okay, and here, I don't know how many faces are there, but there's quite a few. Fun looking at, and the reason I brought this is just to say that, okay, there's a claim that people have specific, they don't have problems with object recognition per se, they have specific problems, gee, I gotta move this forward. They have specific problems with face recognition, okay? And you can see the details are here. Since you're busy with this, we will go on. Okay? Okay. And okay, so they would recognize that there's a person facing them because they'd have something like that. Okay, low pass filter, so they'd be able to recognize there's a face, but they wouldn't know which face is it. Okay? And we were talking about this fusiform face area in that Nancy Canwisher discovered. The question is, okay, is face blindness associated with a dysfunctional FFA? Well, the answer is no. Okay, so I mean, there's different ways in which this has been actually tested. One is to look at the effect of adaptation, or what's called repetition effect. It's called repetition suppression. So if you present the same face over and over again, typically you find that in FFA, as in other areas, you get a drop in the response. So look here, different picture versus same picture, you see that there's a drop of the response for the same picture versus a different picture. I don't want to get into the full details here, but okay, so you get an idea of the repetition suppression in the right fusiform and left fusiform area, and these are the congenital prosopagnosia individuals, so they can't tell a face, and they can't recognize famous images. At the time it was Bill Clinton or Clint Eastwood or whatever, but they still show the same sort of pattern of adaptation that the normal subjects do, although they seem to have somewhat weaker activation. This is only 6 individuals, so it's a question, what do you make of it? But they don't, okay, I think that the researchers here meant to find that there was a difference between the two groups. They don't find this. Okay, 2 minutes about this. What do we make of that? fMRI is a crappy technique, right? That could be, right? It doesn't have the spatial resolution. Wait, so Okay, let's develop this. fMRI is a crappy technique. So, and why? Because if you were if you were to use a more sophisticated technique in which you had higher resolution, what would you find? Leave the sort of repetition suppression, but what do you expect? If you had a higher resolution, you would find that, okay, can you help me? Yes.
Maybe you want to say that we would find that the age-selected cells react in a less resolved fashion, but it's also possible that the FFA works completely perfectly in these people, but that they can't read out from it, or even that in all people the FFA exists but has no meaning whatsoever, and that face recognition is done in a completely different area, not yet identified. So there are many.
Yeah, there are many options. There's no question about that. I think one is which people have later looked into was you encode different faces by the sort of one way to think about it is we talked about distributed coding, right? So actually, if you look at the mean response does not change much, but one face is encoded by one sort of pattern of activity and another by another pattern of activity. I'm not going to talk about it more because Hagai is going to kill me, although he's not going to talk about faces. But that's the idea. That so you will hear about this a lot more from Hagai. And don't worry, I'll interrupt you. We'll discuss this. But that's just a primer, and I don't want to kill Hagai's talk about that. Okay, so the answer here is that was no, but that I don't think that means much. By the way, in recent years, there were developments. I was talking about Yitzhak Fried. There's a group in Stanford called, led by Joseph. Gee, I forget names. Joseph something, okay, who's a neurologist, and they do. I mean, they sort of what they do is use ECOG. Have you heard about ECOG? Mm-hmm. Okay, so with ECOG, what's good about ECOG compared to fMRI? Two things: spatial resolution, temporal resolution, right? Okay, mainly temporal resolution. Okay, so temporal resolution, just like EEG, is at the order of 1 millisecond. Okay, and what they do is they use this in cases of epilepsy, and they they place these electrode arrays subdural from beneath. Okay, so they don't. It's not. I was talking about electrodes which penetrate into the cortex. No, that's not the case here. But they can now use the signals from these. It's basically EEG but subdural, so right on top of the cortex. Okay, and now you have much finer temporal resolution, and now you can, if you sort of focus in on these regions in the occipitoventral cortex, you can suddenly find that you can decode different faces using information based on ECOG. By the way, I think Rafi Malach's student who does similar stuff with Yitzhak Fried was talking about this. I don't know if any of you heard Idan Davidesco. That's what he does. Okay, so that's a very interesting technique. The main limitation is, of course, that you rely on these these patients that are implanted with the ECOG, which doesn't happen very often. So it's a great direction for a PhD, but I think it is problematic in the sense that you're terribly reliant on these very few cases, although they're very interesting to study. Ken?
So you really need to drill a hole, right, to implant those ECOG?
No. So an ECOG, as I said, is something that it's basically an array of electrodes that is subdural, so below the dura, but it's not actually a hole that you do because you come from a ventral side, so from underneath, and they're sort of, if this is the brain, it's sort of covering it from.
So it penetrates the skull, right? It's supposed to.
Yes, of course you make a hole. Yes, okay, but the skull is just knock. There's nothing, right? So really, the issue is, do you penetrate the brain? Okay. So you do penetrate the dura. Yeah, it is subdural. So yes, and the dura is a sort of protective sheet over the brain. But again, the dura is not very interesting in itself. So it is invasive. It is invasive, of course. So it's not used, you know. Absolutely, it is invasive. You do this only in patients. Yeah, that's what I said, of course. Okay. Okay, and just so I'll get someplace, okay, I just want to finish with one thing here. First of all, this is, again, MRI showing this is a sort of a caricature, but this is, okay, work from Uri Hasson, Rafi Malach, in which they sort of found that there's these areas that are sensitive to faces, objects, and buildings. Okay, so this is sort of the atlas again, and this is actually a ventral view and a lateral view. And the point here, okay, so there's selectivity to faces, objects, and buildings. Do we think about this in terms of domain specificity? Well, what about shoes? What about bottles? What about whatever? Okay, so I think it boils down to this issue of a neural coding. Well, should you think about this in terms of sort of distributed versus clustered? And here you see this is sort of at the level of functional MRI, so we're talking at, you know, coarse grain. And of course, you can sort of translate this into sort of a representation which could be either sparse. So again, a representation here. Now you're talking about neurons here, but it could be voxels, of course. Either very sparse. So the idea here is that you have, say, two objects. This and that. Okay? Here's the response to that, object B, in one sort of, so a sparse sort of representation. Here's a response which is across the populations. You see, most cells do respond to this object, different level of firing, while here it's very few cells that respond to that object. And for that object, again, very few cells. So this is sparse coding versus a sort of a true sort of population coding. So which one is the more appropriate one? Well, you'll hear about this a little from Haggai and from others, actually from Dan as well, of course. So I'm not going to go deeply into it. Just say the following. Okay, I mentioned shoes and bottles, right? So, okay, this is what Jim Haxby used at 2001. 8 different categories, scattered ones, and, okay, you can see for yourself. And the idea here was, okay, take one of these categories, say faces, houses, okay? And now you can look at basically a distributed and overlapping representation of faces and objects in ventral temporal cortex. The idea is the following. This is sort of a slice in which you see the patterns of activity evoked for faces, okay? So this is an even runs. Say you have 100 runs. You divide 100 times in which you've shown faces, okay? You divide them to 50 and 50, and look at the evoked response in the first 50, in the sort of the even 50. Okay, so repetition number 1, 3, 5, and so on would go into the odd runs, and 2, 4, 6 would be even runs. And now what I'm trying to basically to show to you is that, look at this. These are the blobs evoked in 2 different sections, section 1, section 2, okay? In the even run, look at this one, and compare this to this one. So immediately you can see that they are pretty much the same, right? You can measure this. That is, where you see a blue blob here, you see a blue blob here as well, okay? And where you see a red blob here, you see a red blob here. What are these blobs? They're the level of activation. So you take the mean activation, say, okay, if it's red to yellow, means it was above the mean activation across the categories. If it was blue, it's below that, okay? So the red ones would be here, face. These are the face blobs we were talking about, because you're showing here faces. And you can show that these activation patterns are quite remarkably similar, and you can correlate them to see how correlated are they. They're 0.81, okay? Again, here, these are a different slice, highly correlated, okay? If you take this is a response to houses. Again, you'll see a stronger degree of correlation when you compare this. But if you compare them across categories, they're anti-correlated, meaning that the areas which are active when you show a face are inactive when you show a house, and the vice versa. And if you do this across categories, you find that, okay, along the main diagonal, this means how similar is the response for a given category in the odd and the even trials. So they're positive. They're all positive. It's better for faces, much worse for scissors. Okay, quite good for sort of scrambled versions. But for faces with houses, I don't know which one is the house here. Second one's the house. It's anti-correlated. Okay, houses with faces, again, anti-correlated. You understand what we're seeing here? You get an idea? If not, you'll hear more about this from Dan. So it's a primer for you. Okay, and basically, you could now use this to show, sort of generate a metric of how similar are the activation patterns for one image category with another image category. And that's what you see here. Basically, there is selectivity for a given image category, and the patterns are very different for other image categories. And basically, you can think about this in terms of a distributed representational object. Again, so here's neuron number, and you can see here the activity generated by one image and another image, and you ask yourself, okay, how similar are these firing patterns? How would you sort of, what's a way to sort of quantify this? A simple way to quantify this? Okay, time's short. It's just to do the correlations. You're saying, okay, here's one pattern. Let's cross-correlate it with another pattern, this one, or correlate it with another pattern and see how, what's the degree of correlation. So that would be, okay, so you can take the activity of, say, neuron number 1 in one condition and in another condition. So that's in for object A and for object 2, object B. And do this for neuron number 2, whatever. Okay, number 3 fires high, so that would be number 3, and so on and so forth. You get this cloud, and you can compute the correlation coefficient of this cloud, meaning how similar is the pattern of activation for object A versus for object B. If it would be ideal, what should be, how should it look like? A perfect line here, right? The dot should be along the identity line here, just to give you an idea. Okay.
Just for one neuron?
No, that's across neurons, right? Each point here is one neuron. So this, say, is neuron number 3, firing highly for the face, right? And also for the object. And this point here would be, I don't know, neuron number 1 firing at low rates for both object 1 and object 2. Okay. And this, you'll hear more about this from
You're making too much expectations.
that's I'm building expectations. That's my okay. You will talk about this. Can I ask you to talk about that? Because okay, I just want to add one last thing because I've just minus 1 minute, so I'll jump over that and to I didn't mention much, but there's one big aspect which I did not talk about. Have you heard about this from Shaul? Reverse hierarchy theory. He mentioned that. And that's the role of the, okay, so he applied to this. And that basically implied, basically, we were talking all the time until now about sort of a hierarchy going upwards, right? In terms of the receptive field becoming more complex and talked about face areas and so on and so forth. And in my introduction, in the first 2 minutes or so, I talked about, the fact that for each basically link from the lower area to higher area, there's also a comparable link, just as strong, a backward, a feedback sort of direction. What could be its role? Any ideas? Okay, so here's what, feedback is what I called it, feedback connections. Okay, so here's the idea that sort of Shaul and Mirav Achisar developed, and that is, okay, that you have this feedback connection to add details to an explicit vision with scrutiny. And the idea was, if you flash an image like this, very short, say 10 milliseconds, and then you mask it. Okay, maybe I have it here. Okay, so you have a stimulus, you flash it for a short period of time, and then you mask it with a masker. Well, you get the gist of the scene, even if this stimulus is shown, the stimulus onset asynchrony, the time between the onset of this image here of the penguins and this masker here is 40 milliseconds. You're still able to tell if there's an animal or no animal in this image. But of course, you don't get the details. You can answer, is there an animal or not, but you cannot tell a lot of the details. So when you have more time, what you can do is use this feedback sort of loop to get more details. And how do you get more details? Through what? Yes. So attention is the key operator here. And of course, you'd be able to tell what this is, but this may be more difficult, okay, to sort of find where does the image, the figure end, and where does the ground start. Okay, here's just one other dimension to it. And I know, my time's up, okay? But so what I want to show you, there's a lot of really interesting stuff here, but who cares, okay? And what I want to show you is that basically you find that if you present a stimulus, and there's multitudes of examples of this. If you present a stimulus in V1, okay, and this is sort of the background, and here you get something against a background in a different orientation. What you find is, okay, and the circle here represents the receptor field of your V1 neuron. What you find is, okay, that this V1 neuron prefers, say, an orientation in this direction compared to that direction. So if you compare, if now the image is such that this is what the receptor field sees, you're going to get a nice response in C and much less response in F. And this is the difference between the two, okay? You follow me up to now? Now, what if you place it instead of here, you place the stimulus such that the receptor field of this neuron is actually covering that area of the visual scene. So you want to compare A and D. Okay, now let's look at A versus D. And what you find is that the initial response is just the same. Okay, but here, what's different in A compared to D? That A is within the figure, while D, there's no clear figure here. So there's no figure-ground segregation here, but here there's a clear figure of this square here, which is not present here, correct? So what you see is that, okay, the initial response is just the same in A and D. It's the same orientation. Everything locally is the same, right, in terms of the receptor field. But if you look later in time, something like 100 or a little more than 100 milliseconds, you suddenly get this difference in the response. And that has to do, okay, with sort of something. So how do I interpret this? How do we interpret this? That something reached higher level, infratemporal cortex, in which you've been able to sort of segregate figure and ground, and then the feedback goes back to V1 and affects its firing such that if this stimulus is within the figure, the response is higher than if it's within the ground. Although the receptor field, as I said, the classical receptor field is the same. So it's not that there's any difference in terms of local difference between A and D. It's just being in A, you're part of the figure, while indeed there's no figure. There's a few other examples on this, and there's a lot to talk about attention. Maybe I'll steal some time in the next 5 to 10 minutes before the lectures, but I don't think I'll do this, okay? Maybe at the end. And I probably won't do this in the end either because you're going to be tired and it won't really interest you. If anyone wants to hear more or something, you're welcome to come and ask me questions. יאללה, להתראות, תודה רבה. Oh, by the way, I sent my lecture to Dan. I'm sorry I did not do this ahead of the time, as I should have. I know I'm really bad at these things. But Dan at least has it now, so have you distributed it? Everyone has. לא פגעתי בהרצאה שלך יותר מדי. הפריימר מותר 10 כן, זה היה מטרה. לכולכם. חגי, מה שאומר דווקא, אני דילגתי שם, בגלל שדילגתי את ההקדמה, אתה יכול לחתוך את זה, אבל את הקטע של הפסקתי שם, זה מאוד חשוב. חלק מההצגה, נכון?
לא, יש לי
אז
יותר
זה הדבר,
מדי.
זה בסדר, אתה לא צריך, אל תדחף. גם לי יש פה, היה מצגת שהייתה אולי 30% ממה שעשית. בסדר, מה חשוב שמה שאתה אוויר שיהיה, כאילו...
אני ממש מתמקד בלהצליח להעביר מסר.
זה טעות.
כן, לא, אני אשיג את זה, את הפיגר מהמאמר, אני אשיג את זה.
מה שאתה בוחר, אני אומר, הפיגר, היה פיגר אחד שלמדתי טוב, אני אציג לך אותו.
כן, אני אשיג אותו.
העניין של ה-vector representation הזה, כאילו דיברתי על העניין של הקורלציה, לא דיברתי על העניין של ה-individual trials. בסדר, לא, זה דווקא נחמד, זה קומפלמנטרי.
כן, אני אראה.
כי זה כן דורש זמן.
I have a question about the face area basically. So we seem to have identified one in humans and in monkeys.
Yes.
What about other animals?
How much work have you done on other animals? In terms of that face areas, what? In rodents, for instance, or what?
Because I can imagine that maybe it correlates somehow with the... Because
I'm interested in humans. Okay, monkeys is my background is in monkeys. Okay, that's as far as I go. You know, rodents, okay, faces.
right, but so as far as why an area like this would exist, like in humans and monkeys are very social, so it makes sense.
Yes,
But
yes.
what about other? I don't know how social rodents are, but
Yeah, I think I would bet that it has to do with social interaction. I mean, there's two elements. First is just recognizing the traits of the face, and then recognizing the individuality of the face.
right.
Okay? And I suspect that, you know, the important thing is, okay, face recognition, you know, this is a lion, this is, you know, this is sort of a predator, or this is prey. That, I think, is something you would expect across the board. But individuation,
Right.
who cares what lion it is? Is it Elsa or Ingrid that's tackling me? I want to know.
Okay, so maybe that's just like the difference between category specific and within the category, then again specific for.
Yeah, but clearly in recent monkeys, they are very social animals. So I think they clearly can individuate. I mean, the reciprocate. There's, you know, heaps of psychology about social interaction. I think that's right. I would bet that, you know, if you what determines if you have these now maybe faces. I was about to say what determines if you're going to have face areas or not is social interaction. But then maybe scent are more important.
Right, maybe it would be
individuality
more like
would be, you know,
a smell
exactly.
area for
Yeah,
a specific
yeah,
person.
yeah. So I would, you know, my guess is that individuality is likely to be based on pheromones and sort of in rodents, sort of they're non-visual animals. I mean,
right, whichever sense you use most. But
yeah.
can you imagine that such a like a specific ability to recognize individuals exists across
Oh,
species?
yes. In species which are yes. There's a hypothesis. I don't know the answer, right? But I think you're right. That I would bet. As I said, I think the faces. I mean, maybe the visual domain is not central in rodents, and the question is how social are rodents in terms of you know mating with a. I just don't know.
Sure.
I just don't know. I bet they don't, but I don't know. I really, you know, you just caught me here in terms of. Adin Mizrahi should know. You should, yeah, you should ask.